Hello, and welcome to Alex, Maran, and Diego's video presentation of DNA microarray technology, which is a useful technique to quantitatively measure mRNA expression.
mRNAs are mature sequences of RNA transcribed from expressed genes in the cell of interest. In order to measure gene expression, we need to isolate these mRNAs.
This is done using reverse transcriptase, which binds complementary DNTPs to the mRNA sequence, creating single-stranded cDNAs. During the creation of cDNA, one of the DNA building blocks used in the synthesis of cDNA carries a fluorescent tag in order to label each cDNA. Different colored fluorescent tags can be used to differentiate between cDNAs coming from different samples, allowing us to compare the quantitative data obtained from different samples.
Of course, DNA microarray analysis would not be possible without the DNA microarray itself, which consists of as many as 1 million different single-stranded DNA segments that are permanently attached at one end to a glass slide or silicon chip. Each DNA sequence is linked to the slide or chip in a known location and serves as a probe for a specific transcript. In other words, each one of the thousands of spots in a DNA microarray contains single-stranded DNA from a unique exon found in the genome of interest.
To create a microarray, thousands of short single-stranded DNA sequences were synthesized in defined positions on a glass plate. The synthesized DNAs represent portions of all exons from a genome of interest.
After cDNAs have been fluorescently labeled, these cDNAs will then bind to the complementary DNA probes on the microarray. This is called hybridization because hybrids between probe DNAs and cDNAs will form.
Then a special scanner is used that shines a laser light on one spot at a time to induce fluorescence. This scanner measures the fluorescent intensity of each spot on the microarray and converts this into a quantitative reading that the researchers can analyze.
We can also use the colors of the fluorescent tags to obtain information about gene expression. For example, if we label control cell cDNA with green and experimental cell cDNA with red, we would see more green in the well of the cDNA expressed highly in the control and vice versa.
A microarray is an extremely useful tool that allows researchers to study quantitative data about the expression of thousands of genes at a time. As a result, they can identify which sets of genes are expressed in concert under specific sets of conditions. In conclusion, DNA microarrays are an excellent way to quantitatively measure gene expression in a cell of interest. Thank you for watching our video.